It's my pleasure to introduce Dr. Gidi Rechavi. He's going to teach us all about the pathobiology, what are the molecular drivers and the microenvironment. And I'm really looking forward to this.
It's a pleasure to be part of this effort to cure follicular lymphoma and to discuss the fascinating biology of this challenging disease. You all know it all starts with the lymph node or similar organs like the spleen and well-there ring, etc. So here is back to the histology class where you see the lymph node. There is the cortex of the lymph node, which is composed of follicles of B cells. This is the B cell zone of the lymph node. In the paracortex, this is the T cell zone of the lymph node. And you see in the right, follicles that are composed usually of resting small B cells. However, upon encountering an antigen, a virus, etc., there is the appearance of these small green spots, 1 to 2 millimeters in size, which are the germinal center. And as we heard from Mitch, the germinal center is the origin of most B-cell lymphoma and follicular lymphoma in particular. You see in the right a primary follicle, a follicle with no germinal center, and then you see the germinal center that appeared again upon activation by oncogene. This is a very famous scientist, Walter Fleming, the one that is the father of cytogenetics. He identified chromosome and chromatin. He described for the first time mitosis and many other things. Among the other things, he also was the first one to identify the germinal center. And at that time, he noticed that the germinal center is composed of cells with high mitosis rate. So he suggested that this is the origin of B cells, of lymphocytes, and from here the term germinal center, which you know this is not true, but he is the one that identified this germinal center. As in closer look, you see the germinal center, which can be divided to dark zone and light zone, surrounded by the mantle zone of the normal B cells that are pushed to the periphery by the growing germinal center. So B cells, as you know, start the life in the bone marrow. They are derived from the hematopoietic stem cells, and in the bone marrow, these hematopoietic stem cells differentiate towards the early immature B cells that leave the bone marrow and reach the follicles, the lymphatic tissue, and there, upon encountering an antigen, they start to proliferate, and this results in the dark zone of the germinal center. Later on, there is differentiation, and the mature cells, or almost mature cells, leaving the germinal center and give rise to memory B cells and plasma cells. Quite complicated process. We have the naive B cell that is coming from the bone marrow. It has a surface immunoglobulin, and there is a large variety of B cells with a huge diversity of surface immunoglobulin. And if one of these B cells recognizes an antigen, then it starts to proliferate, but not only proliferate, there is activation of an enzyme known as AID, activation-induced deaminase. We'll discuss it more later. And there is very rapid proliferation. Actually, the doubling time in this part of the lymph node is about 6 hours, which is faster than any dividing cells in our body. Then those cells that, due to the process of somatic hypermutation, acquired mutation that decrease the affinity of the surface immunoglobulin, those cells usually are dying by apoptosis. However, those cells that, due to the accumulation of mutation, now have a high affinity to the antigen, they are selected during collaboration with two types of cells. One is the follicular dendritic cells that has a processes that present complexes of immunoglobulins, complement, and antigen, and the T follicular helper cell, a subtype of CD4 T cells that both these cells, by interaction with the B cell, provide signal to differentiation and to another process, which is the class switch recombination. The primary B cells always have a surface in IgM. Later on, upon class switch recombination, they can switch from IgM expressing cells to IgG, IgA, IgE, etc. And then the mature cells are leaving the germinal center and becoming memory B cells, waiting for the next wave of antigen, virus, etc. And the long-lived plasma cells that secrete the antibodies. These are the two main processes that result in the huge diversity of immunoglobulin genes. Actually, it's a fascinating system that actually results in unlimited repertoire of immunoglobulins and surface molecules that can recognize any bacteria, any virus, even viruses that we never met. So the first process that occurs in the bone marrow is the VDJ joining. Pieces, gene segments known as the V, D, and the J in the heavy chain genes are recombining in a process that is mediated by the REG enzyme, recombination activity gene. And the end result is a rearranged gene that encodes for the variable region of the surface immunoglobulin gene. Then the cells are leaving the bone marrow, and at the germinal center, another enzyme is activated. This is the AID. And AID is a very smart enzyme. It can do somatic hypermutation. It can introduce mutation at a rate which is 1 million faster than any other cell in our body. And in the light, this occurs in the dark zone of the germinal center. And then in the light zone, there is the class switch recombination, which diversified the heavy chain types of the molecules. So here again, the B cell counters the antigen. There is rapid proliferation, somatic hypermutation, selection for those cells with high affinity to the antigen in the light zone, apoptosis, elimination of those cells with low affinity of the surface immunoglobulin, and production of plasma cells and memory B cell. This is the main activity of the AID, the activation-induced deaminase. As the same indicates, this enzyme in the top, you see a double-strand DNA with C opposite to G. The enzyme can deaminase the C, and instead of C, now we have U, which is not one of the canonical letters of the DNA. Upon this deamination, there is mismatch between the U and the G, which activates a variety of repair enzymes, and the activity of these repair enzymes, it introduces all kinds of mutations, and this is the key for the somatic hypermutation. If these events affect the two strands of the DNA, there is breakage, which is part of the process of the class switch recombination. So this is what's going on in the normal germinal center. It turned out that the majority of, as we heard it in the introduction, the majority of human lymphomas are lymphomas of B cells, and the majority of these lymphomas are derived from the germinal center. So we have a fantastic machinery that can provide us fantastic antibodies and activated memory B cell, etc. However, there is a price for this fantastic machinery, and the price is that all these processes that are associated with introduction of mutations and double-strand breaks, all this predispose us to develop lymphomas. So as you see, and was mentioned by Mitch, many lymphomas such as germinal center lymphoma, diffuse large B-cell lymphoma, Burkitt lymphoma, and more are derived from the germinal center and represents the price that we pay for this fantastic machinery. So follicular lymphoma is a disease of the follicle. Usually the pathology is follicular, not always. However, there is disruption usually of the discrimination between the dark and the light zones. And actually, the germinal center biology is hijacked by follicular lymphoma. This is the key for this disease. And there are a few points to be remembered to the rest of the talk. One is that we have a follicular lymphoma. However, the distinction between the dark and the light zone usually is not there anymore. It's the expression of CD10 and BCL6. This is probably the most important part. There is ongoing somatic hypermutation because those cells are stuck during differentiation at the level of the germinal center cell. They keep mutating their genes. And therefore, and this is probably the major key why it's so tough to cure follicular lymphoma, because patients keep accumulating mutations due to the activity of the AID. Another point that's it's good to remember is that practically you won't find follicular lymphoma where the B cells do not express surface immunoglobulin. And remember, we have a process that all the time introduces mutation to the genes, including the immunoglobulin genes. So you would expect that in some of the malignant B cells, they will lose the surface immunoglobulin, and practically it's always there, indicating that you need signaling from the surface immunoglobulin for a continued proliferation and survival of germinal center cells. These are the early stages of follicular lymphoma pathogenesis. It starts, as you heard before, from the translocation. In about 85–90% of the cases, there is a translocation that brings the BCL2 gene under the control of regions of immunoglobulin heavy chain genes. This results in constitutive expression of BCL2. Then, very similar to the normal germinal center reaction, there is rapid proliferation and selection processes, and then due to the accumulation of additional events, mainly in genes related to epigenetic control, but also other types of mutations, there is blocking differentiation. So the cells proliferate, but are stuck in the germinal center state where there is ongoing somatic hypermutation, and this is the key understanding of this disease. In about half the individuals, and going up with age, using sensitive methods, you'll find circulating cells with the BCL2 immunoglobulin gene rearrangement. So this indicates, and as we heard before, that the BCL2 activation by itself is not enough. This is not enough for lymphomagenesis. So these cells circulate. There are some indications that maybe higher level of these cells is associated with increased risk to develop follicular lymphoma. By the way, the phenotype of these cells is surface IgM positive, surface IgD positive, and CD27 positive, indicated that actually these are memory B cells, and they are there. Only in very rare cases, additional mutation push these cells towards lymphomagenesis. So these are the follicular lymphoma-like cells. Half of this audience probably have some of them in their body. Then the acquisition of additional mutations, mainly again, or in epigenetic regulating genes, but not only, we get the cells that are very important, which are the committed precursor cells. These are cells that acquired enough translocation, and they can expand clonally, and they can give rise to several processes. One of them is in situ follicular neoplasm. In the past, we used to call it in situ follicular lymphoma. Only 5% of those patients that acquire such lesion will go on to develop follicular lymphoma. Usually it's an accidental finding. Then there is partial follicular lymphoma. Only some region of the follicular is involved. Again, not all such patients will go on to develop lymphoma. About half of them, however, will continue to progress towards follicular lymphoma, and then there is follicular lymphoma dissemination, and all this is accompanied by constitutive expression of BCL2, significant expression of BCL6, importantly, continuous expression of the AID enzyme, and the acquisition of additional mutations and genomic instability. The common progenitor cells are very, very important. These are the key to the relapses that we observe so frequently in follicular lymphoma. In the left, you see an example of a patient that diagnosis, a biopsy number 1 was taken and the DNA was sequenced. You see that there are 3 mutations in light color and 1 in red. And then 7 years later in relapse, another biopsy is taken. And you see that there are 3 mutations, including the exact breakpoint in the 14-18 translocation. They are shared by the 2 events. However, there are some unique private mutations that occur during this evolution. This is the key. Probably the cell population of the common progenitor cells is the reservoir that can acquire the additional mutation and result in progression and relapse of follicular lymphoma. On the right, there are few cases that were described of individuals that donated bone marrow to a recipient, and after 3 years in one, 11 years in the second, the recipient, there is development of follicular lymphoma. And when you sequence the follicular lymphoma of the 2 individuals, you see that they have shared mutations, again indicating that in the donor, there were those common progenitor cells that were transferred also during bone marrow transplantation, and over the years, acquisition of additional mutation resulting in independent events of follicular lymphoma. Going back to what we believe is the first step towards follicular lymphoma in the majority of cases, which is the 14-18 translocation. It was described for the first time by the famous Janet Rowley. She described a lymphoma cell with 2 translocations, the 8-14 and the 14-18 translocation. And then in 1984, Carlo Croce, together with the famous Peter Noel that identified the Philadelphia chromosome, they cloned the breakpoint, the joining piece between the 14 and 18 chromosomes, and found this gene, which is called the BCL2. A year later, Cleary and Sklar came with a similar finding in another patient. And then the Croce group found a very interesting finding. They found indication that this fusion of the 14 and 18 chromosomes sequences are due to mistakes in VDJ joining. So you remember this process that occurs in the bone marrow of VDJ joining. In the vicinity of each one of the segments, the V, the D, and the J, we find recombination signals. These are heptamer and nonamer, 7 base pairs long and 9 base pairs long segments, conserved sequences that are separated by a spacer. The spacer can be of about 12 base pairs and or 23 base pairs. And this is the key to the machinery of VDJ joining. What they found is that you expect to have these signals in chromosome 14, where the immunoglobulin heavy chain genes are located. But interestingly, you see that also in chromosome 18, in the vicinity of the BCL2 gene, there is a recombination signal indicating that during B-cell differentiation in the bone marrow, there is abnormal VDJ. Instead of VDJ joining, there is immunoglobulin BCL2 joining. And then, as we heard, Susan Corey and Jerry Adams showed that the first time that this gene, the BCL2, is not a regular oncogene, but is an anti-apoptotic protein. And then the late Stanley Kortmeyer and his group established a transgenic mice that express the BCL2 immunoglobulin genes, and they were able to show that the B-cell survival was prolonged and there is follicular hyperproliferation. And we already heard about 30 years later, the identification of venetoclax and similar drugs. So we have the BCL2 immunoglobulin gene, but as we already heard, it's important, but probably not sufficient. So you need additional mutations. And the era of next generation sequencing now teaches us a lot about the common mutation in follicular lymphoma. So in the majority of cases, you'll find mutations in epigenetic modifiers. Then there is a mutation in immune regulators, mTOR signaling, JAK-STAT signaling, NF-kappa-B signaling, the cell cycle control. And it's easy to remember this primitive scheme that the origin of follicular lymphoma is due to genetics, epigenetics, signaling, and microenvironment. So the genetics is the classic 14-18 translocation with BCL2 activation. And then there is epigenetic, which is very, very important. In the vast majority of follicular lymphoma, you'll find usually more than one mutation in epigenetic regulators. Just what is epigenetics? We have in each cell of our body practically the all 20,000 genes that humans have. So why in liver cell there is expression of one cells, and in brain cell another set of genes is expressed, in malignant cells, in follicular lymphoma, in each cell there is activation and expression of a particular set of genes. This is regulated mainly, not only, by epigenetic regulation. There are at least two levels of epigenetic regulation. One has to do with the methylation of cytosine in the DNA. We have in the DNA A, G, C, and T. The C, the cytosine, can be modified by adding of methyl group at position number 5 of the nucleotide, and this is involving regulation of gene expression. The second type of epigenetic regulation is modification of histones. You remember that the DNA is wrapped around proteins, which are the histone proteins. In this way, when you open the DNA of one cell, it's more than 1 meter long. And then in the cell, it's compacted to 10 micrometer. So the wrapping around the chromosome is very critical. And the compaction of the chromosome has to do with the level of expression, the level of transcription. So there are repressing marks that close the chromatin and prevent active expression of genes. And there are active histone marks and demethylation, which are associated usually with enhanced transcription and gene expression. And as we mentioned, in follicular lymphoma, we find a variety of mutations in those enzymes that mediate and control the epigenetic regulation, mainly at the level of the histones. And there are two important enzymes. One is lysine methyltransferase 2D, which has a loss of function mutations, and this alters the epigenetic writing. And in the EZH2 that we are going to hear about later, there is activating mutation. There is gain of function of this enzyme. And another two enzymes are the CRE binding protein and EP30, which are involved in acetylation of histones. And again, in this case, the mutations are loss of function. There are additional mutations that I won't mention. What about DNA methylation? When we discussed acute myeloid leukemia or MDS, there are frequent mutation in DNA methyltransferase 1, in the TET2 genes, etc. Practically, you won't find them in follicular lymphoma. So for years, people thought that maybe methylation is DNA methylation is not so relevant to follicular lymphoma. However, it turned out that the same AID, this smart enzyme that can do somatic hypermutation and class switch recombination, also can control the level of methylation. And actually, when you know it, it's not surprising, because as I mentioned before, we have the C against G in the DNA. What the AID is doing is turning the C to U. So if there is no C there, there is no way for methylation. There is no substrate for methylation. And therefore, during evolution of follicular lymphoma, you'll see loss of methylation, hypomethylation, and a great variability in the distribution of methylation sites. The third pathogenic mechanism in follicular lymphoma is abnormal signaling. And there are so many abnormal signals that I'm going just to mention it very briefly. For example, the notch signaling, the JAK-STAT signaling that is associated with the mTOR pathway, signaling downstream of immunoglobulin genes, etc. However, I want to remind you what I mentioned before, that always in follicular lymphoma, or practically always, you'll have the surface immunoglobulin, usually of the IgM. So it seems that although there is somatic hypermutation, there is selection for the expression of functioning surface immunoglobulin. And in the last few years, we learned why this is so important. It turned out that due to the somatic hypermutation, there is an introduction in the variable region of the genes, a consensus sequence that result in N-glycosylation, the addition of oligomannose glycans, short sugar moieties, marked here in red, that are expressed at the variable region of the surface immunoglobulin genes. So here is the surface immunoglobulin. This is the variable region. Due to the somatic hypermutation, we have now the motifs for glycosylation, which are aspargine X, serine, or threonine. So there is the addition of oligomannose here. And oligomannose binds a surface molecule known as DC sign, which is expressed on the surface of macrophages and dendritic cells. So there is, in 80% of the cases of follicular lymphoma, intense signaling by the binding of this protein to the oligomannose, which result in intense signaling via the B-cell receptor, indicating that this is essential for follicular lymphoma genetics. Another important gene to mention is the BCL6. Here you see the knockout of the BCL6. When there is no BCL6, there is no germinal centers. And the BCL6 is regulated by a variety of mechanisms, mutations, epigenetic regulation, etc. And its main function, this is a transcription factor that acts mainly as a repressor. And it can repress the expression of many genes. I won't mention all of them. I'll mention only two types. One is a gene known as BLIMP1. Upon germinal center reaction, there is high level of BCL6, which downregulates the BLIMP1. Upon the ending of the germinal center reaction, BCL6 normally is going down and the BLIMP gene is going up. And this is the key to the genesis and differentiation towards plasma cells. So this is usually the end of the germinal center reaction when there are mutations and epigenetic regulation that keeps the BCL2 at high level of expression. There is no differentiation. And this is very important. Another important task for BCL6 is downregulation of genes that has to do with DNA damage response and apoptosis. Remember, in the germinal center reaction, all the time we have somatic mutations, the somatic hypermutation, class switch recombination, etc. Usually when there is DNA damage, there is activation of P53, ATR, ATM, etc. And this can push the cell towards apoptosis. In the germinal center, the key role is prevention of this apoptosis despite the accumulation of lesion to the DNA. So BCL6 seems to be very, very important. And the last important key to the understanding of follicular lymphoma is the microenvironment. We already heard about it. The follicular lymphoma B cells is not alone. It is surrounded and has connection with a variety of cells. Follicular helper cells with macrophages, with regulatory, inhibitory B cells, with CD8 cells, with stroma cells, with dendritic cells, etc. And all these interactions are very important and have two major keys. One, the various cells can enhance the proliferation of the malignant B cells by secreting cytokines, chemokines, etc., by direct connection with the cell. And another role is the prevention of the immune response where the normal immune system can attack the malignant cell. So again, without going into the details, we have a lot of knowledge of alterations due to mutations or epigenetic regulation that on the one hand enhance the support of the microenvironment to the growth of the malignant cells, and on the other hand prevent the immune response against the follicular lymphoma cells. One of the key is downregulation of MHC molecules, which has to do with connection with another cells. The presence of lymphoma cells also change the composition of T cell of various cells in the microenvironment. And as you heard, there are works that showed that the composition and gene expression of non-malignant cells in the lymphoma are associated with prognosis, response to therapy, etc. We already heard that we have an ongoing somatic hypermutation process. Therefore, it's true for any tumor, but it's very relevant to follicular lymphoma. These cells keep accumulating additional mutation. Therefore, even at diagnosis, when you compare the sequence of biopsies taken from two different nodes in the body, there will be shared mutations that usually are derived from the common progenitor cells, but there are also private mutations that are relevant to different regions in the body, and probably we'll hear about it later. Liquid biopsy probably will enable us with knowledge regarding the changes all over the body and not only the one biopsy that we take. The same is true also during the progression of disease. The progression of disease can be transformed or non-transformed. The non-transformed just relapses of follicular lymphoma with the same pathology. Again, there are common mutations, but also acquisition of private mutation. And again, without going into the details, there are the CPC, the common progenitor cells with a specific set of mutations, then additional mutations that can give rise to linear evolution where you have repeated episodes of relapses with the follicular lymphoma pathology, but also divergence evolution where there is acquisition of very active drivers that push the cells towards transformation, mainly diffuse large B-cell lymphoma, but not only, and we are going to hear about it later from Dr. Rossi. So this is just an appetizer. I'm not going to discuss the therapy, just gave the background for the new therapies that we have. I just want to mention that usually when we discuss follicular lymphoma, we start with the 14-18 translocation. However, we know that if there is a family member affected by follicular lymphoma, the chance that close relative is a little bit higher to develop follicular lymphoma. And there were several studies of genome-wide association studies that identified variants in the genome that are associated with increased risk to develop follicular lymphoma. The first two such sites are in linkage with the MHC, it's not surprising. And later on, few additional mutations or alterations that are present in the population can increase the risk of some individuals to develop follicular lymphoma. And the last two slides, as a pediatrician, I want to mention a variant of follicular lymphoma, which is the pediatric type nodal follicular lymphoma. We find it in children, in young adults, but it can be identified in people 40 years of age and older. And it's very important to identify it because the prognosis of this lymphoma, when you compare it to classic follicular lymphoma, is excellent. Usually it's is grade 1. You can just take out the lymph node, you can irradiate, but no more therapy usually is necessary, and the majority of the patient will be okay. Important to recognize it to prevent over therapy of patients. So what are the characteristics of this lymphoma? There is no rearrangement of the BCL2. Mutations in epigenetic regulators are very rare. There is high proliferation rate. The key 67 is higher than 30%. There is low genetic complexity, and their frequent activation of the MAP kinase pathway. So it's very important to remember it. And when you see a young patient, always bear it in mind. This is a major question. There are two explanations that I can suggest, but probably there are more. One is that there is ongoing somatic hypermutation, which probably inactivate the BCL2 at specific cells. And as we heard before, you can live with no BCL2. So BCL2 probably has to do with the initial stages. It enables, actually, there is somatic hypermutation. Usually when there is no BCL2, any accident that activate oncogene, for example, etc., usually will push the cells in the normal germinal center physiology. This cell will be eliminated by apoptosis. However, if you have an anti-apoptotic signal, there is a chance for accumulation of mutations, and later on you don't need it anymore. That's why it's not active. By the way, remember that common progenitor cells, which are the key for the relapses, are very important, and they are still in the very early stage. So probably in the future, when we have in mind effort to cure follicular lymphoma, maybe the time to use anti-BCL2 is very early by using sensitive methods to detect those individuals with specific level of the BCL2 immunoglobulin genes, and then use it for prevention or early intervention instead of using it late when it's not very active.